All right, ladies and gentlemen, welcome back. So we are going to continue our discussion on speciation today and just sort of talk through some mechanisms by which we can create new species. Most students probably think about geographic isolation as the first step to speciation, but that isn't always the case. So when you're thinking about that geographical isolation where some portion of the population gets separated completely by some sort of physical barrier in their environment, what you're thinking about is what we call allopatric speciation. There are situations where speciation can take place without that physical separation occurring, and that's where we talk about sympatric speciation. So let's talk through these two types in a little more detail. So allopatric speciation is really where that gene flow becomes interrupted or at least reduced enough when a population is divided into two or more geographically isolated subpopulations. So the example here with our cute little chipmunks, all right, they used to be a single species living in a single large population over geologic time. All right, this canyon and this river cut through their population. They get geographically isolated. They can't get across that river. Conditions on either side of that river are unique enough to have caused them to diverge because that gene flow was completely stopped. They weren't able to get to each other. They weren't able to mate. And so their gene pools have become unique enough that they are now considered separate species. Clearly, the size of that geographic barrier, the type of geographic barrier, depends on the type of organism we're talking about. So if we're looking at these chipmunks, that river is going to be a clear geographic isolating event for them. They can't get across without dying. They can't swim across that river. That being said, if this were a bird population, this probably wouldn't be much of a barrier at all. They're capable of flying across that canyon, continuing that gene flow, keeping those two populations the same species. In order to determine whether or not that allopatric speciation has actually occurred, not only do they need to be geographically isolated, but eventually they have to evolve to the point of reproductive isolation. So remember, we're talking about the fact that they will no longer successfully mate and successfully create viable, fertile offspring. That's what we're defining reproductive isolation to be. So reproductive isolation between populations generally increases as the distance between them increases. So those barriers to reproduction are intrinsic, meaning that something has happened within their genetic structure, their behavior, their morphology, something to that set of individuals in that population has occurred. It's not just a matter of isolation itself or separation itself. We have to reach that point of reproductive isolation to truly talk about completing this idea of allopatric speciation. So while geographic isolation can be the first step, it is no longer, it is not the final step. We need to reach that reproductive isolation for this to finally be considered two separate species. So that means that in order for that divergent evolution to occur, those separate populations have to adapt to a variety of changing environmental conditions for those two groups, or maybe even more than two groups, to develop into distinct species based on those environmental circumstances. So it's that change in those species due to those environmental pressures that will ultimately produce that reproductive isolation, in which case we can all, at that point, talk about those two, three, or even four separated populations diverging from that original population to become their own separate species. Sympatric speciation, on the other hand, is a lot harder for a lot of students to wrap their brains around because this is going to take place because something else is happening intrinsically within a subset of individuals in a population, but they're still capable of interacting. They're still geographically overlapping populations. So we usually look at sympatric speciation as a result of one of these three mechanisms. Some sort of chromosomal change occurs, so probably some sort of random mutation has happened. Non-random mating, sexual selection, so some change in mating behavior or mating preferences happens in a subset of that population. Or we can get the appearance of some new ecological niche that a subset of this population happens to fill, do a really good job at doing, and then evolve within that new set of conditions, that set of parameters that niche has opened up. So if we talk sympatric speciation from the sense of the chromosomal change, we get this condition of polyploidy, where we've got chromosomal alterations in which the organism now possesses extra sets of chromosomes. And this is going to happen usually due to accidents during cell division. So when we're talking about going to make those gametes, something fails to separate. And so we get to the production of that next offspring who now has an extra set of chromosomes. What I will say is this is rarely the case in most species students think about. They'll think about animals, birds, that kind of thing. Most organisms don't do particularly well with gaining extra chromosomes. But we know what has happened historically in divergence of species, and we also continue to see it much more commonly in plants. Their genetic structure seems to handle this a little bit better than most of the organisms kids come up with off the top of their head. Humans, this is not going to happen for us. All right. We do not deal well with that extra set of chromosomes when it comes to then making that next generation. Think about individuals with Down syndrome. They've got a lot of conditions and cognitive developmental delays, physical differences, so on and so forth, and they've only got one extra chromosome. Most offspring are not viable with extra sets of chromosomes in large numbers, and that's kind of what we're looking at here. So autopolyploidy, just to give you some definitions here, is an individual that has more than two chromosomal sets and they all diverge from a single species. Allopolyploidy is a species with multiple sets of chromosomes that then come from different species. Again, not typically something we see in most higher level mammals or other animals, but plants are pretty capable of handling this to some degree depending on the type of species you talk about. They are particularly common in plants that will propagate or create offspring via asexual reproduction. So if we can get plants that we can graft tissue together and produce an offspring that way, they'll do this. Other organisms that are capable of doing asexual reproduction by putting out runners or tubules or something of that nature. Also, their genetic structure is a little bit more flexible, and so we can get situations where this occurs. And what ends up happening is by having these extra set of chromosomes, we'll often see that a subset of that extra set actually can then, through other random mutations, take on extra new traits that we end up seeing in those offspring. And if those new characteristics happen to be beneficial, they can become more common in the population, and then we eventually end up diverting out into another new species over time. Sexual selection can also drive sympatric speciation. So we're going to go through an example here that your text looks at in cichlid fish. So sympatric speciation in this case is a result of non-random mating due to a particular selection based on sexual characteristics. So those cichlid fish, their selection is prezygotic. It's based on behavior, so it's happening before they even mate. And all of that sexual selection is based on coloration. Interesting thing with this particular example is that this divergence in nature must be relatively new because we can actually pull them into a laboratory setting where we can actually get them to mate and produce fertile offspring if we remove their ability to see that coloration difference. So here's the example. If we were to look at these fish in nature and or in the lab under normal light, we can see that there's this clear distinction in their coloration patterns. And so the females will choose the males based on that coloration. So they seem to have a preference of coloration for their own male that has become so distinct that we now look at them as two different species. And yet, the interesting thing is we can pull this into a laboratory setting where we can sort of whitewash, if you will, um, using orange light, the coloration, and the females can't tell the difference anymore. So it truly is based on color that they're seeing. And they can actually mate with the opposite species, if you will. So females from this species will mate with this male in laboratory settings and vice versa. And they're actually capable of successfully having offspring that are fertile and viable. So that tells us that sexual selection is clearly pushing them towards this divergence point, but it's new enough that if we take it away and they can't tell the difference, that they will actually still be able to successfully mate. So their genetics haven't diverged enough over evolutionary time that they can't actually produce fertile offspring. So again, this brings into that question of that biological species concept. Do we consider these separate species? Well, in this case, scientists are, but it's not really based strictly on their ability to produce offspring because they just won't do it. So it's based really in behavioral characteristics this time around because the females just won't choose the opposite male in a natural setting. So just kind of wrap this up, give you some big headlines from this. So we look at the differences between allopatric and sympatric speciation. Geographic isolation disrupts that gene flow. In allopatric speciation, eventually that reproductive isolation arises because of natural selection, genetic drift, sexual selection, whatever the case might be in those isolated populations. And even if contact is restored between those populations, something is going to prevent the interbreeding from occurring. We get to sympatric speciation, and that reproductive isolation, or that reproductive barrier, is what's causing the isolation of that subset of individuals in the same geographic area. So allopatric geographic isolation starts it, reproductive isolation will end it. If we look at sympatric speciation, that speciation event is occurring because of a reproductive barrier, not because of a geographic one. So they can still intermix, but eventually, because that reproductive barrier is strong enough, they will ultimately become different species, and it can be a result of their chromosomal structure, natural selection, sexual selection, or some combination of all of the above or some of the above, depending on what the situation is.